Good afternoon. Today we'll be presenting the finding of a research paper which was published online by the University of Chicago.
The aim of this presentation is to review the original article and present the implication for the active form of human insulin based on the structure. Although the exact nature of the insulin in this feed is unknown, there is strong evidence that the C-terminal part of the B chain is a dynamic element in insulin activation and receptor binding. Human insulin is identical in structure to your own natural insulin, but when it is injected under the skin, it does work as well as not natural insulin. This is because injected human insulin cling together and take a long time to get absorbed. This shows that the activity of this synthetic human insulin is not well synchronized with your body needs.
Insulin, as we know, can be obtained from beef or pork pancreas glands. There is another insulin called the human insulin. This is made by a method called semi-synthetic or recombinant DNA. Insulin is a protein hormone which regulates blood glucose level, resulting in the metabolism of protein and lipid. Insulin is composed of two peptide chains known as chain A and chain B. They are both linked together by two disulfide bonds. Chain A consists of 21 amino acids, while chain B 30 amino acids. The first picture is of an insulin dimer. This is formed by hydrogen bonding together. The second picture is that of an insulin hexamer, and this is formed by the presence of zinc. In the process, insulin dimer deassociates to form insulin hexamer.
Uses of insulin in the treatment of diabetes. Insulin's metabolic insulin receptor. Insulin receptor is a tyrosine kinase. It functions as an enzyme that transfers phosphate groups from ATP to tyrosine residues on intracellular target cells. Binding of insulin to the alpha subunit causes beta subunits to phosphorylate themselves, thereby activating catalytic activity of the receptor. The activated receptor then phosphorylates a number of intracellular proteins, which in turn alter activity, thereby generating a biological response.
Looking at these chains A, B, and C, as you can see presented on this slide, is to identify the main structural changes in insulin during a transition from the ground state to a proposed activated active form based on the structures of the highly active analogues. For the structure A, it shows an overlay of the general fold of crystal structure, while the location of the B26 turn is marked by the star, magenta, while structure B shows that the clause of B of B26 turn in the representative analogues, which is nitrogen and oxygen atom, in blue and red, respectively. In C chain, it shows how superposition occurs.
This slide is about the molecular shape of active insulin. It can also be said to be the characteristic of active insulin. Active insulin has some characteristics which include 1. Insulin activation at the phenylalanine B25 and B26. Another of its characteristics is that they have hydrophobic knobs, and that another one again is that they are highly hydrophobic in their IR binding cleft. They have different relative orientation
Molecular shape of active insulin. Insulin mRNA is translated in the beta cells of the isolate of Langerhans in the pancreas, generating 110 amino acid precursors called preproinsulin. Preproinsulin is a relatively inactive hormone. By the removal of a signal peptide during insertion into the endoplasmic reticulum, and with the addition of 2 disulfide bonds, preproinsulin is converted to proinsulin, and within the endoplasmic reticulum, proinsulin is exposed to several endopeptides to cause the 31 amino acid C peptide chain, which then derive insulin.
For this slide, material and methods. These are some of the methods that have been to show these analogues and AC. Crystallization conditions were initially found by an in-house developed screen and were refined by the handing drop method. The structures were known by molecular replacement with differently truncated monomeric insulin.
Finally, the understanding of the activation of human insulin will help in the development of a unique type of hormone analogues in the treatment of diabetes mellitus. The normal insulin B25 to B30 is in close contact with other A and B chain residues in the ground state of insulin molecules. Thank you for your attention.